Peace be upon you all, and welcome to this installment of Seeing God in Biology. And we are still in the central dogma. And today we will extend the discussion that we did the last time for transcription in prokaryotes, now into eukaryotes. Fuz, how are you doing today?
I'm doing great, Ahmed, and we couldn't get enough of transcription, so we have to do it in 2 episodes. But the last time we talked about the concepts and the principles of transcription using prokaryotic organisms, simple organisms, as a way to kind of elaborate those concepts. And now in this particular session, we're going to look at how transcription happens in eukaryotes, and we'll see similarities, but we'll also see some significant differences that reflect really the vast difference in complexity of eukaryotic organisms compared to prokaryotic organisms.
Yeah, so last time we stopped at the point where DNA was really converted into RNA, and now I think this RNA will be maybe processed in a different way in eukaryotes. So let's jump back into your presentation, and everybody, let's see Dr. Fuz Rana tell us how the system can get even more complex. Yeah.
Okay, so now we're going to move to eukaryotic systems, and the level of complexity is now amplified when we start talking about eukaryotic genes. These are the types of genes that are found in organisms that are made up of cells that have a nucleus. So these are complex cells, and the genes typically again have a similar to a prokaryotic gene, a promoter region where the RNA polymerase binds. But what we see is that within the coding region is that there are these sequences that interrupt the coding sequences. These are called introns. And so a gene will consist of exons, which are the coding regions, and introns, which are interrupting non-coding sequences. And so the coding information is dispersed along the DNA, where it's interrupted by intronic sequences. Okay, so once the gene is transcribed, those intronic sequences are removed through a process of splicing, where the sequences are cut out and the exonic sequences are joined together to form what's called the mature RNA molecule that then directs protein synthesis.
Was it at a point of time that geneticists thought that introns were just junk?
Yes, that is the sense that introns are junk, but there's now discoveries that indicate that once the introns are spliced out, and those are they're spliced out in the form of what's called a lariat, and we'll talk about that in a second, that the lariats are actually performing functions inside the cell. And I just read a paper about that, and at the moment the function escapes me. But we're discovering that those intronic sequences after the fact are actually functional. But as we'll see in a minute, there's a reason why you want to interrupt the exonic sequences with introns, because it creates greater degree of versatility in terms of what kind of transcripts can be encoded in a single region of DNA through a mechanism known as alternate splicing. And so alternate splicing wouldn't be possible if you didn't have these intronic sequences. So they are minimally serving a function in that capacity.
I actually wanted to ask you this question for another reason, which is, again, the why question. So it just strikes me every time that when evolutionary biologists, which you can extend to evolutionary microbiologists and evolutionary biochemists and evolutionary anything, when they see something that they don't understand, they just jump into conclusions so fast and say it's junk, it's waste, it's a fossil of the inefficient process of evolution. It's evidence that this system is like haphazardly developed. But if they were really teleologically thinking, they would have asked the why question, and it would have driven them into more inquisition and more inquiry, to the extent that they would have progressed science much faster, right? Rather than just waiting for decades and then finding then what they have called previously junk is completely not junk, and there's so many uses of it. So once again, a system that is designed will always answer the why question. And the teleological mentality, this is another example that you've just given us, where the teleological mentality, the design-oriented mentality, will progress science much better than the blind natural selection / random mutation driven mentality. Would you agree to that?
Oh yes, absolutely. I couldn't have said it better myself. It's there's a the consequence of the way you think about biology is profound. Where if you are approaching it from the standpoint that there's a mind behind everything, you're going to seek after purpose. And if
Yeah.
you don't, then you're going to be very quick to conclude that things which with purpose actually may lack purpose. But when you think of, when we look at the splicing mechanism, what we'll see in a minute is that splicing mechanism is extremely precise and is extremely complex, and it's an energy intensive process. And so, if these introns were truly junk, natural selection would evolve or force genes towards uh, or create the evolutionary pressure to eliminate the intronic sequences, not to have them in the sequences. And so
That is just presupposing or assuming that natural selection can do anything of the sort. But I guess you are actually using the logic of Darwinists against them, that you cannot actually have your cake and eat it in the same time. You cannot say that natural selection will eliminate energy consuming processes, and then when you find something that you don't understand, you just call it junk, while you know that this junk is really consuming so much energy. So it's paradoxical to give these answers. However, they just keep giving them instead of just saying that we are ignorant to the magnificence of the design. But and this is where somebody's philosophical stance can hinder him from actually accessing the real knowledge. Would you agree to that? Do you think?
oh
What do you
yes,
think?
yes. I think that's extremely well stated. And so so often I hear the charge that if you say that God is responsible for some feature in biology, that it shuts down scientific inquiry. Well, not really, because you and I both think that God did it, and we have enormous amount of motivation to study it more so because we want to see the Creator's fingerprints. We want to gain a glimpse into what God has done. It's kind of tongue in cheek a little bit, but it's like we want to think God's thoughts after Him.
Yeah.
We want to appreciate the beauty of the creation in as much detail as we can, if for no other reason than it just prompts us to worship and to celebrate, God, who is the Creator.
So
And
I'll just ask you this question. Let's assume that we are back to Paley's time, and biology is based on teleology, right? And I'm just for the fun of it. So Dr. Rana is, responsible for approving papers, right, or peer reviewing them. And then somebody writes in a paper that we have found that there are introns, and my position is that introns are junk. And then you will immediately not approve this thing and go, don't just throw your ignorance at me and tell me it's junk. Go and do your inquiry, or just write in the paper that we don't understand yet what it's used for, and this is an area for further study or further research. This would be your position. Now, if I am on the other side, back to that time, and the leading position is Darwinism, I would just accept the paper and call it a day and accept introns as junk, and hence I would have killed possible new research.
Yes.
And it turns out over and over again that the teleological position would be the correct position, and the Darwinist position would be the lazy position that hinders science and hinders progress. And
Yes.
and this is what real research proves every day.
Yes. Yeah. And eventually, what has been labeled as junk or as a vestige, a non-functional vestige of evolution, will eventually, scientists will eventually discover that function, but usually it's not in direct pursuit of the function of that system, but it happens to be just a coincidental discovery, or that discovery, it happens in spite of the paradigm, not because of the paradigm.
Yeah. Great. So one more confirmation that believing in God and believing in his wisdom and his magnificence and his omniscience, essentially even, drives science forward, not the other way around.
Yes, exactly.
All right. So it was a little bit long interruption, but I think we're really doing this to marvel God's creation and to see God in biology, and to see that believing actually drives you higher and drives you forward and drives you faster and drives you better and drives you more efficient and makes you more intellectually inquisitive and makes you less lazy and makes you less hand waving. It makes you exactly the in the right position to pursue science and to find great discoveries. Not really the way Darwinists or naturalists or philosophical naturalists or methodological naturalists would really like to describe who they call creationists.
Yes.
It is essentially totally untrue and totally unfair.
Yes.
All right. So thank you for giving us the opportunity to interrupt you in this point. And I think this will really raise plenty of questions. So I'm just encouraging the audience just to throw your questions at us for the after show. Yeah, go ahead.
Now, here's, we'll continue. This is showing another twist to the gene structure in eukaryotic organisms, and that is, in addition to the promoter, there are sequences called enhancer sequences. But what's interesting about enhancer sequences is they are not immediately adjacent to the gene itself, but they occur at vast different distances away from the actual gene, but yet they are controlling gene expression. As the name implies, they enhance the expression of the gene. And it turns out that we now understand how that works. And this is a cartoon showing the tan or the orange promoter in structure, the pink RNA polymerase II bound to it. And then we see the enhancer sequence at a distance away, but the DNA double helix is folded to bring the enhancer in close proximity in three-dimensional space to the promoter sequence. And then there are these transcription factors and other proteins that bind to both the enhancer and to the RNA polymerase. And that binding actually enhances the expression of the gene by probably enhancing the binding of RNA to the promoter.
Wow.
Or, sorry, the RNA polymerase to the promoter.
We actually have a video for that, by the way. So we'll
yes.
show it.
Yes. And so the video is worth, yeah, a video is worth 1,000 pictures if a picture is worth 1,000 words. But this is, again, an added complexity, to the way in which the gene expression and gene regulation takes place, which gives you incredible versatility, in terms of, controlling the expression of genes. And you can have tissue-specific enhancers and tissue-specific transcription factors so that the same gene can be expressed differentially from tissue to tissue in complex multicellular organisms.
Wow. So I see enhancer and super enhancer. What's this super guy doing?
Yeah, well, the super enhancer is essentially a situation where you've got multiple enhancer sequences that are in close proximity to each other, so that when the DNA chain folds and brings the enhancers in three-dimensional proximity to the promoter sequence, that you can actually have multiple transcription factors and bind to both the enhancers and to the RNA polymerase, which even further, exaggerates the enhancing effect. So it's just if a little bit is good, more must be better approach.
And at the end of the day, the enhancing effect will cause, what's its eventual effect?
It increases the amount of RNA that is transcribed, and that in turn will then lead to more copies of that protein that's encoded in the gene to be produced inside the cell.
All right. So one more like dimmer kind of protocol to really produce more of the same thing, because maybe it is more appropriate for that kind of tissue or organ or whatever. Yeah.
Right. And here the transcription factors are in a sense functioning in an analogous fashion to the repressors and the activators that we would see in prokaryotic cells, where through the use of enhancers, you can create, a number of feedback and feed forward loop mechanisms that are working in an integrative fashion. So it really creates very sophisticated systems that are regulating gene expression.
So it strikes me that prokaryotes and eukaryotes, there is a certain concept, or there is a certain design that needs to occur. The mechanisms look completely different, but the effect is the same. So it's like, may there be dimmer switches or something, okay?
Yeah.
and then it's implemented this way in that kind of organism, and that other way in that other kind. But essentially, it again gives the adaptive nature and and this control system, but in a different way. That cannot be evolutionarily related, I guess.
Yeah, you're right. I mean, it's similar concepts that are, which is why we can study E. coli or other bacteria and develop an understanding of the concepts. And then with those concepts in hand, as we look at eukaryotic cells, we see the same concepts manifested, but the precise mechanisms may be different. But and this is the reason for this is complex, and we won't get into it. But you cannot have complex multicellular organisms like fungi and plants and animals built from prokaryotic cells. You have to use eukaryotic cells to build those kinds of organisms. And of course, once you're talking about multicellular organisms that have different types of tissues that are interacting to form different types of organs, where there is a growth and development process, now what's required in terms of gene regulation far outstrips just simply, are you looking at housekeeping genes or inducible genes that are responding to something in the environment that single-celled organism may either want to be attracted to or avoid or take advantage of or what have you. Now we're talking about a level of gene regulation that has to be that is exponentially more complex. And what you're looking at are corresponding mechanisms that implement those same concepts, but in a way that accounts for the complexity demands of a multicellular organism.
Amazing. Once again, if you know that there is a designer and you see that he is using certain concepts in one type, and you know that the other organism is even more complex, then you are looking for a more complex implementation of the same concept, because you believe in one designer who has created them all. And if you don't, then maybe you don't even look for the mechanism to start with.
Yes.
So it's not only just believing in the grand designer of it, but that he is actually just one. There is only one creator, one designer for everything, and he just created them all. So once again, more of seeing God in biology, I guess.
Yes.
Yeah.
And then this is, we talked about again the splicing process, and where the introns are removed and the exons are joined together. And so this is a diagram that just gives you an inkling of how complex this process is,
Wow.
where
Wow.
the different colored symbols are representing different proteins and riboprotein complexes that are working collaboratively to carry out this splicing. And essentially what the end product is that the two exons are joined together and the intron is cut out in the form of what's called a lariat structure. And so what ends up happening is you have a very precise sequence in the 5' region of one exon and in the 3' region of the other exon. And and then there's a precise sequence or a consensus sequence in the middle of the intron called the branch point. And what happens is that this branch point is attacking the intronic region right before the exon, and then that once that takes place, then the two exons are brought next to each other, and there's another another attack that takes place. And as a result, the two exons are spliced together, and that intron is released as a lariat structure. So wow. It's a bit of a complex mechanism, but the point is that the sequences that are right at the 5' and 3' end of the splice site, as well as in the branch site, are very precise sequences that cannot be manipulated or mutated, because if they are, you lose the capacity for that splicing reaction to take place. So this is a very high precision mechanism that is removing these introns. And so again, if you're thinking about an evolutionary origin to life and an evolutionary origin, how on earth are you going to evolve, these and introduce introns into a gene to create exons and introns, where once you introduce that intron, it can't just be randomly dropped into the gene. It's got to be dropped in a precise location where the integrity of certain sequences in that intronic sequence have to be very precisely maintained. And then it takes this very elaborate machinery to remove the intron, right? All of this has to evolve simultaneously. This to me is beyond, I think this is beyond reason to think that something like this could have evolved in any kind of, through any kind of evolutionary pathway, other than a pathway that would be totally guided by a creator, or something like this has to be instantiated all at once. And so this also, the complexity of this to me also suggests there's got to be a reason why the cell is going through all of this effort, in order to perform this splicing mechanism, right?
Because at the end, maybe if this is all not needed, just having all the exons lined up in sequence would have been much more efficient, right? And here you go, just read the gene and produce the RNA and go ahead and transcribe it, no sweat.
Right, but what this is telling us is that there's probably this becomes essentially a point where you can begin to regulate or control gene expression, or where alternatively you can do interesting things like alternate splicing, which we'll talk about in a minute. But just kind of as a little bit of a housekeeping bit of detail, this is what now once the splicing has been done and we produce the mature RNA, you wind up with not only the coding region, but also, again, a 3' untranslated region, which is playing a role in positioning the transcript at the ribosome. There's a 3' untranslated region that's playing at minimum a role in releasing the RNA from the ribosome. You also have other modifications. There's a capping process where you cap the RNA, and this capping takes place before the splicing takes place. So the cap is playing a role in the splicing process. It also helps to facilitate the positioning of the messenger RNA at the ribosome. The poly A tail is probably adding stability to the messenger RNA, though it's not clear to my knowledge what the precise role is. And then also in these untranslated regions, there are what are called microRNA recognition sites that actually are also playing a role in gene regulation. And we'll talk about that when we get to the episode on the design of pseudogenes.
Yeah.
And so we'll just put a pin in it for right now. But other than to say that there's again a rationale for the complexity of the RNA structure in eukaryotes that compared to prokaryotic, mature RNAs. And then last but not least is a slide that we want to finish with, and that has to do with alternate splicing, is that because you have multiple exons and multiple introns, you can actually do some very interesting things where you splice out certain introns so that you can actually, with a single transcript, produce, a wide range of alternate transcripts.
Wow. So just correct me if I'm wrong about what I'm seeing here. So these are the colored ones are essentially the exons, and the white ones are essentially the introns. So after taking out the introns, you will have the blue, the red, the green, the yellow, but those can be combined into, for example, in this example, 3 different possible stitching ways. So you can have the blue, the green, the yellow, or the blue, the red, the green, the yellow, or the blue, the red, the yellow, and each one of them, if translated, will essentially produce a different protein.
That's exactly right.
And I guess if this was the whole function of introns, this would be an interesting function, because what you're telling us here, that the same gene can actually produce a multitude of mRNAs and hence a multitude of proteins, depending on how the splicing would really process it, right?
Yes.
And this actually strikes me because I'm not sure if this would be related to that or not, but I would expect it to be, because at a point of time, and actually until now, there is always this, guys, humans are like, great apes, and there is like 90-something% similarity with chimps, and they just keep varying the numbers, 97, 98, 99, whatever they want to push at it. And it's a false number as far as I'm concerned, because there is at least 5% different in length anyway. But I have read the papers, and in the papers it says, regardless the 90-something similarity in DNA, the real proteins are only 30% similar between humans and chimps. So the same genetic, if it was even like 100%, we don't care, because at the end of the day, what makes a difference on the ground is the proteins. And if you compare all the proteome, I guess we can call it, of the human versus the chimp, it's only 30% similar and 70% are different. And you're showing us here a mechanism where the DNA can be almost similar, but just the splicing mechanism will reassemble the RNA in a completely different way, that the genome expression is completely different.
Yes.
Is that it?
Yes, that's right. And this creates enormous amount of versatility, so that the same gene, if you will, can produce different types of transcripts and different types of proteins, and you have a mechanism now to regulate gene expression, as you're pointing out, and to vary the gene expression and the types of transcripts that are produced from cell type to cell type, or at different points during the growth and development process. And so it's, through the alternate splicing mechanisms, again, you create this, versatility, but also you introduce points where, again, very sophisticated gene regulation can take place.
Actually, two parables are popping into my mind as we speak. So the first one is like, when I used to write code, a good programmer would typically put comments in the code. Okay, so on the hard disk, on the hard drive, the C code or whatever kind of language he's writing in has the real code, and there are always the comments. And the comments are very important because otherwise a different programmer cannot read what's happening. And when the compiler or the interpreter reads the code, it will essentially forsake the comments and just focus on the code. But it doesn't mean that the comments are junk. It doesn't mean that the comments are not useful. And maybe geneticists will have an objection to that because they will say, well, there aren't different programmers reading, and we don't need comments here. This is an execution system. And hence, but it just reminds me of the way we humans write code. We don't just write the code. We have to put those extra stuff in there so that the code is meaningful. But then the second parable comes to my mind. So let's assume, and correct me if I'm wrong, if the parable is really good or not. Let's assume that the gene is essentially a sentence, and that each one of those exons is essentially a word in the sentence. Now, let's now envision those other elements of the sentence that we call punctuation marks, okay? And the punctuation marks on their own, they have no meaning at all. But a full stop, a comma, a semicolon, a dash, parenthesis, an overstrike to cancel actually one of the words can really cause the same 6 or 7 word sentence to have so different meanings, to the extent even using the punctuations smartly, you can actually produce exactly the opposite meaning from the same set of words. And so somebody cannot just come, let's throw out just all the punctuations. But to me, just one use, and I guess this is what you have explained in alternative splicing, that those introns are essentially punctuation marks that's telling the system, here are the words that are encoded in the DNA, but now let's use the punctuation marks to produce really different meanings according to what we want. So we can have genes that are so similar between chimps and humans, but the author of this book wants it to spell humans, and of that book, he wants it to spell chimps, and he just needs to change the punctuation marks. Part of it is changing the punctuation marks. There's a whole other lot of it. But this is language, and this is very sophisticated programming system that is using a very efficient system so that you can express the 4 words in this example to produce a multitude of 4-word sentences and 3-word sentences and 2-word sentences and 1-word sentences. And would you agree that this parable can actually work? Or what do you think of this?
Yes, I mean, I think it's very helpful to think of, these think of biochemical systems as information systems, as and apply information theory and information concepts, because I think the analogy is a very powerful analogy that really helps us to make sense of what's happening here, but also gives a reason why, or suggests reasons why this would be the structure of genes in eukaryotic organisms. Because it creates a versatility that and a complexity that is concomitant with what's needed to produce complex multicellular organisms. And you are right, that what differentiates humans and chimps in a significant way are the gene expression patterns that you see in the tissues. And this alternate splicing is part of the gene expression patterns, that you would see in humans and chimps. It produces different proteomes that are, cell specific, tissue specific, and maybe even, organism specific.
Yeah, and actually it strikes me that every cell in our body has 100% similar DNA, but you can get brain cells and liver cells and kidney cells and bone cells that look completely nothing like each other, and they're the same DNA, but it's just expressed in a different way. So I remember having a debate with a fellow theist who was actually Dr. Joshua Swamidas, and he was referring to the similarity, etc. But then I just say that, well, does the similarity really count if you can really produce so diverse outcome out of them using small tweaks? And it only takes a genius writer, a genius author, to give him the same set of words and to ask him to produce two completely different stories using as minimum as possible changes to the words that you would give him. And isn't it this really what we say? So I would really say that the similarity that produces huge diversity is a hallmark of an ingenious designer who uses the same electronic components to produce, on the one hand, a laptop, and on the other hand, a mobile phone. Wouldn't you agree to that?
Yes, yeah. Well, yeah, the concept of modularity, is very powerful, and the exons are most likely encoding regions of protein structure and function that allow that in a sense could be thought of as being modules that are combined in different ways to make functional proteins. So you see that modularity of design in using the same elements but over and over in different and unique ways to create a wide diversity of systems. And there's one of the things that many theists marvel at is the intelligibility of nature, right? That it's one thing that we live in a beautiful, complex universe, and that we see in the universe beautiful, complex organisms, but it's another thing that we can actually understand them. And part of that, at least from a Christian perspective, and I'd be interested from an Islamic perspective, the idea is that, well, as humans, we bear God's image. And so in other words, God designed us in such a way that we would have the mental capacities, the cognitive function to discern and understand the world, so that part of the intelligibility lies within our capabilities, but part of the intelligibility lies within the way that God has made the universe and the systems that are part of the universe, including living systems. And so when you have modular designs, not only is that an elegant way to design, but it's an elegant way for someone to understand the designs. All I need to do is understand how a particular electronic component works. And then when I'm confronted with a new design that's made up of a combination of electronic components, because I know how those work together and how you can combine individual components to produce even more complex modules, that I can begin to study that new design, even if I have no idea what it does, and begin to make sense of it, right? And so I think, to me, God wants us to see his fingerprints and his handiwork in the created order, and creating these modular systems is, the way to go.
Yeah, actually, it's also pretty interesting that man created in the image of God. There's actually a hadith in Islam. Prophet Muhammad says, خلق الله آدم على صورته. God has created Adam on his image. Some have not understood the hadith, and they said, okay, he means his image. It means that God created Adam in Adam's image, that he has been created like Adam all the time. There has not been a chimp that was an ancestor of Adam. But I think that it can also be understood in the same way that God has created Adam with similarities to the qualities of God, because God is merciful. He wants us to be merciful. God is wise. He wants us to be wise. He gives us the ability to be wise. We can choose not to be wise, of course, and be arrogant and ignorant and stupid and everything. But we can also be wise. We can be gracious. We can be grateful. And all of those are supreme qualities of God. So I think I personally believe there is a metaphorical also aspect of the hadith that God has created humans in His image, in the same sense that you're talking about. And there is also another Islamic concept, and I'm sure we'll find something in the Torah or the Gospel in the same meaning, that says سنريهم آياتنا في الآفاق وفي أنفسهم حتى يتبين لهم أنه الحق. We will show them our signs in the horizons and within themselves until it is evident to them that it is the truth. So it's pointing humanity to look into two places: to the frontier of their knowledge, which is the way I explain the word horizons, obviously including the real horizons, to look into telescopes, to look into deep space, look as far as you can. And within themselves, I personally look at it as look to the very deep micro level of your own creation, to essentially what we're doing today.
Yeah.
And then you will see that it is the truth.
Yeah.
And the truth is His word is the truth, and He is the creator. And it ends, the ayah, this is an ayah in the Quran, it ends, أَوَلَمْ يَكْفِ بِرَبِّكَ أَنَّهُ عَلَى كُلِّ شَيْءٍ شَهِيدٌ. Isn't it sufficient for your Lord that He is over everything witness?
Yeah.
So He knows this, and He knows that. He knows how our amazing cell biology is working. It's just telling us, go and look. And when you look, you'll find my signs. And this is actually the reason I personally have a deep belief that asking the why question for a person who believes in God, we're not afraid to ask the why question. We want to ask the why question to find His signs that He has asked us to find. So definitely, God's revelation, whether in the Torah, Gospel, Psalms, will point us to the same direction. Look at the horizons of your knowledge, look in yourselves, look into the creation of animals and plants, and you will find me. And I've given you the tools. And I think, Fuz, what you're doing in this amazing series is looking into ourselves, because essentially humans are the pinnacle of God's creation in terms of living things, and we are eukaryotes, right? We have those very complicated structures, right?
Yes.
And they are manifesting themselves in the most amazing way, where we can essentially do what you're saying. We have the ability to understand an intelligible universe, including intelligible life. It's really amazing. So I think this is. Thank you for doing this, and I'm sure many of the believers who are watching this, it brings tears to one's eyes and just so illuminating. And I hope some of those who are agnostic would lean to the other side now, to the faith side, and that some atheists to really reconsider, because this does not look like a universe that has come by any other means rather than by intentional and purposeful design. There is no other solution, because I would really not use politically correct language, that it is more reasonable than not that this is really design. It's there is actually one solution. This is design. This is a very big is, with a capital I and capital S, I guess. What do you think?
Yeah, well, to paraphrase Richard Dawkins, that where he defines biology as complicated things that give the appearance of having been designed for a purpose. Well, maybe if it appears to have been designed for a purpose, it actually was designed for a purpose. and I think that's really what we're seeing in each of these episodes, is that the designs in biochemical systems that we're really only in the last few decades have really begun to understand and to appreciate are just are screaming design in ways that I don't think we could have ever anticipated or even fully appreciated.
Absolutely. So thank you very much for this super exciting episode. And for our audience, join us on the other side on the after show. And thank you very much, Fuz. And I'm really looking forward to the coming the installment coming up, which would be about now translation, right? This
Yes.
spliced RNA will now magically transform into proteins, right?
Yes.
Yeah, so this will be super fun. So thank you very much, Fars, for this. Great insights and great teaching. And thanks for everybody who's watched this. And peace be upon you all. And see you in the after show.